Hey class, so we're talking about the integumentary system right now, and in this recording, we're going to be focusing on the epidermis in particular. So as we look at the epidermis, I just want to emphasize some root words. Epi means above. What is it above? It's above the dermis. And this epidermis is going to be made of a keratinized, stratified squamous epithelium. Stratified meaning multiple layers. Squamous meaning flattened cells. Keratinized, which means that they are going to be filled with keratin. And the most superficial layers, or the apical region of this tissue, is going to consist of dead cells that are going to be totally filled with keratin protein, but the basal region of this tissue, deeper in the body, is going to have living cells. It's worth emphasizing that this is an epithelium, and because it's an epithelium, it's going to be avascular. It lacks blood vessels. So to get nutrients for tissue growth and cell growth and maintenance, we're going to depend upon diffusion from the underlying connective tissue of the dermis, which the epidermis sits directly on top of, or directly superficial to. The epidermis itself, it has very, very few nerve endings for touch and pain. And the reason for that is because the epidermis is, for lack of a better term, the sacrificial lamb, or the sacrificial layer of tissue designed to be destroyed to protect, well, it's protecting the underlying layers of tissue in our body. So as we look at our epidermis, there are 5 cell types within our epidermis. We have a bunch of stem cells. These stem cells are going to be located in the deepest layer of the epidermis, the stratum basale. And this particular kind of stem cell is commonly referred to as an adult stem cell. Technically, it's known as a multipotent stem cell because it's been partially differentiated. I want to emphasize that there's a lot of really cool research going on right now, which harvests multipotent stem cells from the skin of an individual and then reprograms those multipotent stem cells to act like pluripotent stem cells. And then this field right now is starting to develop is using this particular kind of stem cell from the skin to grow custom organs that genetically are a perfect match for the recipient. I could go on for a very long time, but we're going to shift and we're going to focus on the epidermis right now. The single most common cell type in the epidermis is the keratinocyte. Those stem cells are primarily going to turn into keratinocytes. To review the root words, cyte means cell. Keratin refers to the fact that they are filled with keratin protein and make keratin protein. So the overwhelming majority of cells within the epidermis, by default, when you look at a cell, just assume it's a keratinocyte. It's the vast majority of cells within our epidermis. We also have some melanocytes. Cyte again meaning cell. What does a melanocyte do? It makes melanin. It synthesizes melanin, and I want to emphasize there's the IN at the end of this word. This is a pigment. A lot of students will get melanin mixed up with a neurotransmitter called melatonin, which plays a role in regulating our circadian rhythm within our brains. So I just want to emphasize melanocytes make melanin, the pigment, not melatonin, the neurotransmitter in the brain. Melanocytes will be stimulated to make melanin when we expose them to ultraviolet radiation, or when we expose them to, you'll love the name of this, melanocyte stimulating hormone. And the purpose of this melanin is to block that ultraviolet radiation from damaging the DNA within the cells of our skin. We have the melanocytes only within the stratum basale of our skin, and they have very branched processes. So they kind of have a dendritic or branching or tree-like appearance to them. And those branched processes allow for them to send the pigment melanin to many nearby keratinocytes. And those nearby keratinocytes absorb the melanin and become pigmented. Within our skin, we also have a lot of tactile cells, and these tactile cells are the touch receptor cells, and they're typically going to be in that deepest layer, the basal layer of the epidermis, known as the stratum basale. And these tactile cells near the surface are the light touch cells. So when you feel somebody rubbing a feather across the surface of your, gently rubbing a feather across the surface of your skin, or maybe a light, gentle breeze, that's something that you could pick up potentially and feel from a tactile cell. Although I personally would argue that it's the hairs that are being moved, the hair shaft above the surface of the skin that's wiggling, that you're really feeling, not so much these tactile cells. And we don't have a very high concentration of them just because we don't want to be overstimulated and have constant pain. We also have a lot of dendritic cells, and these cells, in terms of gross morphology, or gross appearance, I should say, when you look at them under a microscope, and I know that's kind of an oxymoron saying gross appearance under a microscope, but when you look at the physical structure of these dendritic cells, they physically have a similar structure to the melanocytes. So they have these branching extensions coming from them that look like the branches of a tree, hence the name dendritic cell. The reason these cells are distinctly different from the melanocytes is that these are cells that are going to phagocytize pathogens. They are a form of macrophage that is exclusive to this epidermis. And they're going to be found in the stratum spinosum and stratum granulosum of our epidermis and aid in our immune response. So here's our epidermis in more detail. You can see that we have lots of cells here in the epidermis. Most of these cells that I'm circling right now in this big green, we'll call it an oval, those are all keratinocytes along different stages of development. So within the stratum basale, this deepest layer, let me shift to reddish here, this deepest layer of cells, the single layer of cells is the stratum basale. The keratinocytes are active and alive. We also are going to have the stem cells here as well. Stem cells are being represented right here. And you can see mitotic spindles, and this looks like you can see a metaphase plate showing that the stem cell is actively going through mitosis and cytokinesis, so they're constantly reproducing. We also are going to have that tactile cell as a sensory receptor, and you can see there's a yellow nerve fiber in this figure going to the tactile cell. We have a branched melanocyte right here that's present, and that's going to be the pigmentation cell. And then I'm going to shift to yellow and make my stylus a little thicker. And then over here, circled in this big, thick yellow line, we have a highly branched dendritic cell. So those are the cells of the epidermis. I want to clear the screen. I want to focus on the strata, or the layers of the epidermis. We have the stratum basale. The stratum basale is the deepest layer. It is 1 cell thick. Then we have the stratum spinosum, which tends to be one of the thicker layers. It's the next deepest layer. And in the stratum spinosum, the keratinocytes are still living. Up next, and I'm shifting to red here, we have the stratum granulosum. And this is when the keratinocytes are starting to fill with keratin granules. This figure shows the stratum lucidium as a lightly pigmented layer directly superficial to the stratum granulosum. And then we have our surface layer of completely keratinized, totally flattened keratinocytes, which are now dead. And that surface layer is called the stratum corneum. It's made of fully cornified or fully keratinized keratinocytes. So I know I stole a little bit of my thunder here, but let's go over those layers again in a little bit more detail now that I've shown you them on the figure. So we have thick skin and thin skin. Thin skin is only going to have 4 layers to its epidermis, 4 strata, and I love saying strata with a really bad German accent, 4 strata to the epidermis. And then thick skin here has 5 strata. It has all 5 layers. So we have the stratum basale, that deepest layer. The stratum basale, to review again, has the stem cells. It also is going to have the melanocytes and some of those tactile cells, which I pointed out in the previous figure. And I need to emphasize it's a single layer of cells. Then we also have the stratum spinosum. The stratum spinosum is multiple layers of keratinocytes. These keratinocytes are bound together by cell-cell junction, we talked about previously, called a desmosome. We're also going to have a lot of tight junctions. The tight junctions add a waterproofing feature between the cells, and the desmosomes are going to give us a rigid, or I shouldn't say rigid, but a structurally, a high amount of structural connection between them. So it's going to serve to really anchor these cells together. So we have a strong cell-cell connection anchoring them, and we also are going to have some cell-cell connections that are waterproofing the stratum spinosum. It's called the stratum spinosum because some individuals believe that these cells have a spiny appearance in photomicrographs. I personally haven't seen this in over a decade of working with these cells, but if you talk to a histology buff, those individuals may disagree. Well, I shouldn't say may. They will probably disagree with me on that statement. There will be a few dendritic cells within the stratum spinosum, the deeper layers of the stratum spinosum, but those dendritic cells are typically going to be concentrated within the stratum basale. Next, we have the stratum granulosum. Oh, went too far there. And as I just to reiterate, when we look at the stratum granulosum, that's going to be that layer, that darker purple layer in a photomicrograph where you start to see those granules filled with keratin. And it's one of the thinner layers. It's typically 3 to 5 layers thick. Our next layer is the stratum lucidium. And I want you to circle, star, underline this right here. It is found only in thick skin. This is a key characteristic. Stratum lucidium is only in thick skin. And the reason we call it the stratum lucidium is because it lets more light through. This word right here, luci, refers to light. I personally always think of the name Lucifer, which is a name that refers to the angel of light. And in this case, this layer is going to let more light through the photomicrograph, hence the name stratum lucidium. It tends to be a brighter layer, and it's directly superficial to the stratum granulosum. There'll be a protein called I'm having a tongue twister here. Elidin. There we go. Sorry about that. Elidin. And that protein is going to help to improve the phototransmission of the cells in this layer. And then finally, we have the stratum corneum, which is the most superficial layer. Depending on the individual, if it's thick skin versus thin skin and lifestyle habits, it can be just a couple layers thick. So think of the skin on the epidermis of your eyelids or palpebrae. You almost never have frictional forces exposed to your eyelids. Thank goodness. That would be really uncomfortable. So we have just a few layers of cells to the stratum corneum in our eyelid. But then think of those people with really thick, cracked heels. They can have 30, 40, or sometimes even 50 or to 100 layers of cells. But those are typically a homeostatic imbalance situation because their heels are so thick, the epidermis is so thick that the heels start to crack and bleed. And that's not necessarily the best thing. As we think of the stratum corneum, I want you to think abrasion resistance. I also want you to think of waterproofing. And I also want you to think of penetration. It's a sacrificial layer. We get little tiny cuts in our stratum corneum all the time and never even notice it. So I just want to back up here and just reiterate, we have the stratum basale as the one layer of cells. We have the stratum spinosum as multiple layers of cells. The stratum granulosum is going to be purple, or it's going to have dark purple granules in it. The stratum lucidium is going to have a lighter coloration, and then we have the stratum corneum, which is going to have the fully keratinized dead skin cells on the surface. So as we look at a keratinocyte, our keratinocytes have kind of a journey they go through their life cycle. Initially, the keratinocyte will be made via mitosis and cytokinesis from those multipotent stem cells that are within the stratum basale. And those keratinocytes are then going to continue to be pushed superficially up through the stratum spinosum. actively reproducing our cells and going through mitosis and cytokinesis requires a lot of nutrients. And those nutrients are going to diffuse from the underlying dermis. Once those keratinocytes get pushed away from the dermis, we can no longer have mitosis occurring and the rate of cell division drops off. So we have cell division and new keratinocytes made in the stratum basale only. Over time, those keratinocytes will be flattened out. They will be filled with keratin and some other vesicles that are going to help to contain waterproofing and antimicrobial proteins. It's going to take about 30 to 40 days for a keratinocyte to make it all the way to the surface of the skin and then exfoliate off as dander or dust. And so like right now, I made this recording in early March, and we just had our first warm snap of the year this last week, and I was able to open up the windows of my house, dust, get all that dead skin, all the nastiness out of my house. And it's kind of cool, but kind of gross all at the same time. If you want to see this process in action, wait till it's sunny and you can have the rays of light shining in the window and hold your arm out in the sunlight, your forearm, and just scratch it aggressively. And you can see dead skin floating up into the air. As we age, this process slows down, but if we have any kind of damage or distress, we're going to accelerate the process. We'll often have calluses or corns form on our feet or our hands because of repeated stresses that we expose to our feet or hands. For example, if you play guitar, a lot of times you get calluses on your fingertips, or if you're a thumb sucker, a lot of times you get calluses on the posterior of your thumb from your incisors constantly rubbing against the skin of your thumb. Next slide. As we look at the stratum granulosum, the keratinocytes are going to release, will have a granule known as a keratinohyaline granule, and it releases a protein known as filaggrin. Filaggrin is going to take those individual keratin proteins and bind them into tough little bundles. So it's going to help to bind and mesh the keratin proteins together. And this again occurs in the stratum granulosum. We're also going to have some envelope proteins, cell envelope proteins being produced just deep to the cell membranes of the keratinocytes within the stratum granulosum. We're also going to have some lipids that are released, and these lipids will spread out over the superficial layer of the cell, allowing for the cell to be waterproofed. And then finally, within the stratum granulosum, organelles will be destroyed and we'll have keratinocyte death. So at the stratum granulosum, we then form the water barrier. And we're going to have lots of tight junctions. And I want to emphasize again, tight junctions are a cell-to-cell junction that waterproofs the cell. And these tight junctions will occur and be formed in the stratum granulosum. And if we don't lose water through our cells or through our skin, it helps to reduce the rate of dehydration. It doesn't prevent the absorption of water by layers of cells that are superficial. So for example, the stratum corneum is superficial to the stratum granulosum. And you probably had this happen where you took a bath, or maybe you went swimming for a long time, and after soaking for a long time, your thick skin in particular that has a thicker stratum corneum has absorbed more water and has become kind of pruny and wrinkly looking. So we can still have water absorption by more superficial layers, but the keratinocytes within the stratum granulosum are waterproofed and prevent water loss. That's all we have for this recording. If you have any questions or comments, please feel free to post them in the class discussion board or to shoot me an email. Happy studies.